Good morning. I want to start, of course, by expressing my most sincere thanks to the organizing committee for giving me this wonderful opportunity. It is a great honor. I am deeply grateful. The letter of invitation stated that we ask that you frame your work in a broader context and do not present only your own work. I have taken this advice to heart, as you will see. But at the same time, I will delve into fields that I am not an expert on, particularly, as you all, most of you know, I'm not a molecular biologist, and so I may make errors in vocabulary, but I hope that I will be able to convey the general principles accurately. I have no conflicts of interest related to this talk. What I want to talk about today is the strong interactions between sleep-wake regulation, the circadian system, and their implication for metabolic health. A similar lecture could be given for their implication for the immune function, for the implication for cardiovascular function. But I think metabolism has a privileged relationship in that particular triangle. In the first part of the talk, I will discuss the novel concept linking sleep-wake regulation and the circadian system. When the first meeting of APSS was held, these were the concepts that were dominating, on the one hand, the sleep field, and on the other hand, the field of circadian biology. These were largely seen as independent fields. It's no longer the case today, luckily. So the two-process model, and we just acknowledge Circadian and Webb as well, posited that across the entire 24-hour cycle, the level of alertness is regulated by two separate processes. Process S, which conceptualizes sleep-wake homeostasis, and process C, which posited that an alerting signal is emitted by the circadian pacemaker to counter the building pressure for sleep across most of the daytime. Process S and process C were seen as both influencing wakefulness, alertness, but really as separate processes. On the right side, you are seeing a cartoon that I had the privilege to develop with Professor Ashoff, widely seen as the father of human chronobiology. And with Ashoff, we tried to summarize some of the major principles of underlying the mammalian circadian system as it was seen at that time. The most important part, perhaps, is that it had been recognized that the suprachiasmatic nucleus of the hypothalamus was holding a circadian clock, of which the mechanisms were unknown, and it was believed that the circadian clock was the one and only pacemaker that was capable of generating a self-sustained near 24-hour oscillation in the mammalian system. It was recognized, of course, that the light-dark cycle was the primary environmental synchronizer, Zeitgeber, would Ashoff say, to the clock. And there was also recognition that social cues and physical activity could also play a role. It was recognized that some organs, and particularly the adrenals, were capable of generating damped circadian oscillations that were only sustained by the continuous entraining input of the central clock. All other organs were considered as passive slaves of the circadian pacemaker, and generating overt rhythms that were entirely dependent on the SEN clock. As these notions were central to the mammalian system, there was effort in the field of genetics of Drosophila to identify the mechanism responsible for circadian rhythmicity in the fruit fly. And our president already saluted the Nobel Prize given to Jeff Hall, Michael Rosbach, and Mike Young. I was recently at a meeting of the SRBR, and Young and Rosbach were there, and the excitement was enormous. The prize is validating, is giving a stamp to the field of circadian rhythms, but also of sleep, a stamp of validity, a stamp of importance, and it is an opportunity on which we must capitalize and feel really energized to move forward. In short, the mechanism generating circadian rhythmicity in the fruit fly is a feedback loop, a negative feedback loop by which the protein product of the gene period inhibits its own transcription. And to do this, the protein needs to be bound to another protein called TIM from the timeless gene and re-enter the nucleus and inhibit their own transcription. The prize was saluted by the Nobel Committee with a statement that has some important points. It says the clock regulates critical functions such as behavior, hormone levels, sleep, and metabolism. It talks about jet lag. And then there are also indications that chronic misalignment between our lifestyle and the rhythm dictated by our inner timekeeper is associated with increased risk for various diseases. The work in the fruit fly gave a blueprint to explore the mechanism underlying the mammalian clock. And this led to the current understanding of the mammalian circadian system. And so in this cartoon, I have summarized what are the main principles of today's understanding of the mammalian circadian system. First of all, we are still thinking that the clock in the hypothalamus in the SEN is a master clock. The mechanisms have been elucidated and also involve a loop of negative feedback. The identification of the major components, the major genetic components of this loop, made it possible to examine peripheral tissues to see if those same genes were rhythmic in their expression. And lo and behold, in almost all tissues examined carefully so far, a self-sustaining clock was present and could be seen in vitro for as long as the cells could be kept alive. And so now we have a multi-oscillatory system with clocks in peripheral organs, clocks in areas of the brain outside of the SEN. The clock in the brain in the SEN is, of course, still seen as primarily synchronized to the external environment by the light-dark cycle. But the clocks in peripheral tissues, particularly those sensitive to nutritional cues, have been shown to be also sensitive to the timing of food intake. And this I have represented in the American fashion with a big hamburger. And you can see there the GI tract and the liver and the muscle, the adipose tissue have not represented the pancreatic beta cells, but they certainly do belong there. in every model system where a misalignment between the clocks in peripheral tissue and the clock in the brain has been produced, or experimentally produced, an adverse metabolic effect was observed. And so in this cartoon, all the clocks are showing the same time. And this is therefore the cartoon of a healthy, metabolically healthy circadian system. So looking a little bit closer at the transcription translation feedback loop, the first gene, mammalian gene to be identified as involved in clock mechanism was named actually clock, was discovered by Martha Witter-Turner in Fred Turek's lab, and then subsequently cloned by Joe Takahashi. Clock associates with another gene, BMAL, and together they promote the transcription of two families of protein, PER and CRY. And there are 3 PERs and 2 CRYs. PER and CRY proteins move to the cytoplasm where they form complexes in association sometimes with other proteins. And then they feed back as a complex to the nucleus to inhibit their own transcriptions. And so this is very similar in the principle to what was shown in the fly. If you look at the rhythms of expression of BMAL and PER in healthy humans, this is this insert on the left lower side of the slide. It's showing you these rhythms across the 24-hour cycle, each of number of healthy subjects studied at the University of Surrey by the group of Dyke, were studied with normal sleep-wake cycles, sleeping approximately from midnight to 8. And you can see that BMAL increases during the day to peak in the middle of the day, whereas PER peaks in the middle of the night. And they're 12 hours out of phase, and this is all very nice. But how does that feedback loop generate a near 24-hour oscillation, constant day after day in all of these tissues? What makes this feedback loop special? That the oscillation that is generated is not 5.1 hours or 3.2 hours, or what makes this loop really generate 24-hour rhythmicity? And I really wanted to understand that better before standing here. And I went to look at papers that had done mathematical modeling of this transcription translation loop, and went to meet a senior author and say, well, how did you find the time constant? How do you know that adding all of these elements of the loop will finally result in a constant 24-hour period? He said, well, you know, the parameters, the time constants are chosen as plausible, educated guesses. And really, we know the components of the clock, but we don't know the cogs that link them. Very recently, at the suggestion of Chuck Zeisler, I attended a remarkable talk at SRBR by Chuck Weitz, another Chuck of Harvard Medical School. And what he was showing is that, and reminded me, he also used, I don't think it's an old-fashioned technique that, as Chiara would refer to it, but he used electron microscopy to basically image the size of the complex, the PER-CRY complex, when it's in the cytosol and when it comes back into the nucleus. And what his work, so he basically is taking a photo, an image of the size of these complexes as the day progresses and as they go, they are being formed. And what the work shows is that these complexes are not simply the purrs and the cry and this additional one that I can never remember how to describe, but that there are additional proteins that form much bigger macromolecular assemblies that take time to produce. And when the complex involving purr and cry finally moves back in the nucleus to begin to inhibit clock and BMAL, 30 more proteins are needed to finally saturate the clock and BMAL and stop the production of purr and start again the positive limb on the loop. So in the words of the authors, the last sentence says, our results show that from the onset of negative feedback, about 7 hours are required for the nuclear purr complex to accumulate to a concentration sufficient to saturate clock BMAL. So now we have a long delay there to build up the molecular assembly, and we have, of course, a similar delay to deassemble. And that explains partly why the period cannot be 3 hours or 5 hours, why it has to be a long period. It's a major, I think, step forward towards understanding not only the components of the clock, but the cogs that make them interact. Now, coming back to the full human body, how does the central circadian pacemaker keep the symphony orchestra of the multiple oscillators in non-SEN tissues, including peripheral tissues, in harmony? And really, the conductor has 3 batons. The first is a hardwire system, is really the partial control of autonomic nervous system activity, which innervates basically every organ of the body. And that represents a hardwire system by which central timing can be communicated to peripheral organ. Second, there are 2 major hormonal signals that are linked to the central pacemaker and propagate throughout the peripheral circulation. One is the rhythm of pineal melatonin release, the hormone of the dark. Melatonin has receptors in the SEN and in the reproductive axis, but it also has receptors on metabolic organs, including the pancreas. And so by that way, the pancreas, in a way, sees the light through the stimulation of melatonin receptors on these cells. The other pathway, which is probably even more important, is the wide, enormous circadian rhythm of circulating glucocorticoids, which was first identified in the 1950s by Pincus. And this rhythm is probably playing a very important role as an internal synchronizer because there are glucocorticoid receptors practically on every cell. And so that high cortisol of the beginning of the active period, the low cortisol of the end of the active period is communicated to all of the peripheral organs. Lastly, there's a behavioral cue that plays an enormous role in keeping the circadian system in harmony. And that behavioral cue is actually the sleep-wake cycle. Sleep itself is an important synchronizer of the mammalian circadian system. And we first stumbled on this, and at the time, the concept of circadian misalignment was not really used for hormones and for rhythms coming from different internal organs. It was more describing a mismatch between external periodicities and internal periodicities. So in this slide, it's from 2004. It was published at that time. It's not discussed much, but I like to show it to you because it's looking at the rhythm of plasma leptin. As you know, leptin is produced by fat cells, and it's a satiety signal that informs the brain about the caloric status of the body. And the slide is looking at the relationship between the leptin profile and the cortisol profile under 3 sleep conditions in the same subject. One condition where the subject were fully rested in dark blue on the right side. One condition where they had 8 hours in bed and achieved on average 7 hours of sleep. And one sleep-restricted condition where they had only 4 hours of sleep. Other than the amount of sleep, everything else was constant. Namely, the middle of the sleep period was constant. The timing of the meals were constant. The meals were identical. The light-dark cycle was identical. The lights were drastically dimmed in the hours preceding and following the sleep restriction period. And despite that, you can see that when the subjects were fully rested, the leptin peak falls exactly at the same time as the cortisol nadir. But on the left side, when the subject had only 4 hours in bed, there's almost a 5-hour difference between the leptin peak and the cortisol peak. So there is circadian misalignment between 2 internal rhythms, and it's only in the last few years that we have started to describe it as an example of circadian misalignment. Wonderful colleagues around the world have now used omics technology to examine the impact of sleep duration and sleep timing on the organization of the circadian system. Series of studies came from the University of Surrey. I'm showing here a study where the same subjects were provided blood samples around the clock, once when they had normal nocturnal sleep and once when they were 12 hours out of phase due to participation in a 28-hour forced desynchrony protocol. And you can see, without my giving any details, that mistimed sleep created havoc in the blood transcriptome, with the amount of transcripts that remained rhythmic decreasing from 6.5% at baseline to 1% during forced desynchrony. So mistimed sleep, as occur in shift work, is a major desynchronizer of the mammalian system. And I'm sorry, this is really, I think, important, is that this slide focuses specifically on the major player of the circadian pacemaker, the transcriptional activator BMAL and the transcriptional repressor PER. So those are the 2 main players of the clock. And we're looking at peripheral blood cells. And what you can see is that when the subjects were aligned, the expected phase relationship between BMAL and PER was clearly there in blue. And when the subjects were submitted to circadian misalignment via the 28-hour forced desynchrony protocol, the rhythms were essentially flattened. So in conclusion of that slide, mistimed sleep affects molecular processes at the core of circadian rhythm generation, not simply circadian control genes. Following this use of the transcriptomics, the team of Ken Wright at the University of Colorado just published a paper looking at a human plasma proteome, sampling around the clock every 4 hours, about 1,100 and some proteins, and noting that about 50% of them had a rhythmic pattern, with 30 of them having an extremely strong, robust rhythm. And that is rhythm, and those are represented on these polar graphs by the turquoise areas. And you can see that when the subject underwent circadian misalignment, which was only 2 days in the laboratory, where both food intake and sleep were 12 hours out of phase, the abundance of robustly rhythmic proteins decreased remarkably. So now what I've talked about until now is how sleep affects the circadian system, which is a view that certainly was contrary to what we were thinking of 20, 30 years ago, because we saw sleep as an output of the clock. Everything I've been talking about in the last few slides is how sleep affects the mammalian clock system. Now, let's think about the other part of the concepts that we had 30 years ago, which is that sleep-wake homeostasis was seen as a process S, that was largely independent of circadian timing. So is that still the case? Well, it turns out that there's also been findings that went against this concept. And the first one was the finding by Lapowski and Naylor in Frederick's lab that the clock mutant mouse slept an hour less than the wild type. And so here, a prototypical circadian mutant has an abnormality in sleep. And using now not mutations, but polymorphism in humans, the Surrey group examined phenotypic differences in a circadian marker, the 24-hour profile of melatonin, and in a detailed phenotyping of sleep, including baseline sleep, sleep quality, and recovery from sleep loss. And the polymorphism is a variable number tandem repeat polymorphism in the PER3 gene, and this polymorphism is fairly common, such that it was possible to recruit subjects who were homozygous for the long version of the polymorphism, and others who were similar in every way, except they were carriers of the short version. And what this slide is showing is that slow wave of all the rhythm of melatonin was completely the same in these two groups of people, despite the fact that they had a major difference in the PER3 gene. But what was mostly affected was sleep-wake homeostasis and slow-wave activity, in particular at baseline and following recovery from sleep loss. So when this paper came out, I was really blown away, because it shook so many things that we were very strongly believing in. Lastly, again at SRBR a couple of weeks ago, there was a talk by Ketima Paul, a student, a professor now, but a mentee of Joe Takahashi and Fred Turek. And he presented data that are really a revolution. They need to be confirmed. So his model is a mouse. There are 4 colors on this slide. I only see 3 from here, but there are 4 colors. There is a wild-type mouse in black. There is a mouse shown for which the data are shown in white, and this mouse is a knockout of the BMAL gene in the entire body. And then some mice were rescued for BMAL in the brain only, and some mice were rescued for BMAL in the skeletal muscle only. Those manipulations are possible now. So what is on the abscissa is non-REM slow wave activity after recovery from sleep deprivation. And you can see on the left panel that the wild type recovers about almost 20% more sleep than at baseline, more slow wave activity than at baseline, but that the BMAL KO in the whole body does not recover. And that, in a way, is consistent with the two-process model. But when the BMAL, the clock, is rescued in the brain, it does not correct the phenotypic defect. On the right side, it shows you when the clock is rescued in muscle only, then the recovery of slow wave activity following sleep loss is recovered. So this is the title of the paper is BMAL1 function in skeletal muscles regulates sleep. And we now have to think about the possibility that peripheral clocks may actually affect sleep regulation, another revolutionary concept for our field. So in the take-home messages from this first part is that the molecular basis of mammalian circadian rhythmicity is not fully understood, and that we can look forward to major findings regarding the cogs of the clock. Sleep is a behavioral synchronizer of the circadian system. It may be the most important behavioral synchronizer. Insufficient sleep has profound adverse effect on circadian organization, as has mistimed sleep. Conversely, genes that are pivotal components of molecular clocks affect not only sleep timing, as we used to think, but also the homeostatic control of sleep. Process S is not independent of process C. Clocks in peripheral tissues may well play a role in sleep regulation. And the translation of this is that sleep health may promote circadian health and reciprocally. I now want to move to the implications of sleep-wake regulation, circadian system for metabolism, and I'm going to talk about metabolic health, about obesity and diabetes. But before moving into that, we have to recognize that the modern times, the contemporary times, are associated with a lot of novel behavioral factors that can affect the integrity of both sleep regulation and circadian rhythmicity. So we have these new lifestyle factors that contribute to induce circadian misalignment and to induce sleep disturbances. We are exposed to light at night, not only white light, but a lot of blue light from these screens that we love so much. We sleep as little as possible, so we crash during the daytime and sleep during the daytime, and to avoid that, we take a lot of energy drinks. We have a 24-hour society with a lot of individuals working irregular working schedule with irregular sleep-wake cycles. And lastly, there's another novel behavior that has been recently documented very carefully by the team led by Sachi Panda at the Salk Institute. And the team developed an app by which people can report the timing of their energy intake, whether it's a drink or whether it's a meal or a snack, by taking a photo, and the photo is sent to a central website. And in this way, rather than relying on paper reports of meal timing and content, this becomes a very much more accurate way. And what you can see here on the right side is that the paper that they published in Cell Metabolism in 2015 already had 11 people participating in this collection of every intake by photo, and they were each studied for 10 to 15 days. Now, if you look at subject number 1 in pink, you can clearly distinguish breakfast, lunch, and dinner. But if you now look at subject number 10 in green all the way to the right, and the subject next to it, well, it's constant grazing. There is no true, other than the lunchtime, it's eating nonstop. And a lot of the subjects were eating well beyond 9 p.m. I drew a horizontal line at 9 p.m. So so thinking about the peripheral oscillators that are sensitive to metabolic cues, this kind of behavior is another way to induce circadian disorganization. And ongoing research is examining the role of extended overnight fast and fixed meal timing in the integrity of the circadian system. So obesity has been increasing in this country and worldwide relentlessly. On the right side, all the prevalence in all the states of the United States is shown for every state between 1919 and 2016. Roughly, the prevalence of obesity has increased 2.5-fold. On the left side, you see the map of the prevalence in the United States. The darker states have a prevalence of frank obesity, that is BMI 30 and over, of above 35%. Only Colorado, Maine, and DC are under 25%. Obesity is a disease that involves, that has a lot of medical complications, and some are shown here on this slide. Of course, we know very well about sleep apnea and snoring, heart disease, liver disease, cancer, and also diabetes. And I regret that the cartoon does not include depression, because that is also a frequent comorbidity of obesity. Diabetes is also becoming extremely common in the United States and worldwide. There's now almost 30 million people in the United States who have diabetes, and an additional 86 million have so-called pre-diabetes, where they're no longer normal glucose tolerance, but not yet frank diabetes. The estimation here is that 1 in 3 will develop diabetes within 5 years, but more recent numbers are more like 1 in 2 will become diabetic within 5 years. And so that's a tremendous epidemic. Diabetes has dreadful complications. It's the main cause of blindness. It's a primary, very important cause of renal failure, of stroke, of heart failure, and lastly, the main cause for amputation of lower limbs. So let me then review the data linking these two images produced by the CDC. Similar images, of course, mean in no ways a causal relationship, but there is ample evidence that, in fact, these two phenomena are related. And I cannot take you through all of the experimental and epidemiologic data. And so here is a summary, and I will illustrate some of the points. So the first thing that it was recognized is that insufficient sleep has an adverse impact on the hormonal regulation of hunger, such that it promotes the release of hunger that the release of hormones that promote hunger, and it inhibits satiety hormones. Insufficient sleep results in increased hunger and appetite by self-report, and imaging studies of sleep-deprived subjects show increased activity in regions involved in food salience and a reward. Insufficient sleep promotes hedonic food intake. Hedonic food intake being food intake above and beyond what is needed for the caloric requirement of the individual. And this hedonic food intake is very often in the form of snacks and can be as much as 300 calories per day. Insufficient sleep also affects food preferences. And lastly, a series of studies has shown that the stimulation of energy intake far exceeds the energy cost of extended wakefulness. So this is an important point because when at the beginning of these studies we noticed a decrease in leptin levels with short sleep, one of the reviewers stated that it may be a normal adaptation to the increased need of calories due to the energy cost of extended wakefulness, even under sedentary condition. That was a very valid argument. The cost of extended wakefulness has now been carefully measured in studies led by Ken Wright at the University of Colorado in calorimetry rooms. And in a couple of studies, they found the same number, and that number is kind of disappointing for those who like to restrict sleep and not gain weight, because the cost of 1 hour of more wakefulness under sedentary condition is only 17 calories. So with 1/2 or 1/4 of a chocolate chip cookie, you have paid 3 hours of extended wakefulness. So looking at some of the data, this is representing the two hormones that have been most often studied and for which the findings are consistent. Increase of ghrelin, and of course the short sleep condition is in red, and decrease of leptin. I want to mention two things. One is that the change in leptin is only seen with a more drastic sleep debt. Here we're looking at 7 days of 4 hours in bed versus 8 hours in bed. And the other point is that you really need to sample across the 24-hour cycle to see when the effect of sleep restriction is actually the most significant. And many studies have only sampled morning fasting blood. So another hormone that we are focusing on now in my laboratory under the leadership of Erin Hanlon, who unfortunately could not be here today, is endocannabinoids. So endocannabinoids are the endogenous ligands of the same receptors as the receptors for cannabis. They exist in two forms, CB1 and CB2. The CB1 receptor has been the target of anti-obesity pharmaceutical effort, and one drug developed by Sanofi Aventis, rimonabant, was on the market in Europe and was very successful in reducing weight, but it also reduced an hedonic drive for things other than food and involved an increased risk of suicide and was taken off the market. Here we're looking at endocannabinoids, and believe it or not, in all of these efforts, multi-million or billion-dollar effort, I don't know, to develop an anti-obesity drug based on this system, the time of day impact on the activity of the system had never been looked at. And so on this slide, Erin Hanlon measured the levels, the circulating levels of the most abundant endocannabinoids, 2-AG. And you can see the black curve is under normal sleep condition, that 2-AG levels, which in a way represent hedonic drive, are low during sleep, and then begin to rise across the morning, peaking at lunchtime, and then declining to returning to evening levels. The amplitude of the rhythm is very large. During the night, the nadir is at 50% of the mean. During the day, it's at 150% of the mean, so it's a very robust rhythm. When subjects are sleep deprived, the peak of the rhythm is amplified by more than 20%, and the high levels persist longer towards the evening. So this would suggest that the drive for hedonic feeding is indeed amplified by sleep deprivation, and that at the same time, this drive for hedonic feeding persists later in the day, consistent with a tendency of sleep-deprived subjects to eat more as the day progresses, evening eating. The lower part of the slide is showing you the ratings of hunger. The left part says, how hungry do you feel right now? And the right part is how much, I can't read that, but it's a similar, how much do you feel like eating, something like that. So the questionnaires are administered just before the breakfast meal, 2.5 hours after the breakfast meal, just before the lunch meal, 2.5 hours after the lunch meal, and just before the dinner meal, and again. And what you can see is that the red and green and black curves are superimposed in the morning, but that they begin to diverge significantly around lunchtime, specifically when 2-AG is higher when people are sleep deprived. So in reviewing the literature on the risk of weight gain and overeating associated with sleep restriction, the work from the University of Pennsylvania, led by Dr. Spaeth, in the group led by Dr. Dinges, has been really remarkable, because this is a very large set of data, and was reported in 4 consecutive papers, and it has told us a lot about vulnerability to sleep restriction. So large cohorts of healthy adults, 20 to 50-year-old, non-obese, studied under control condition of the laboratory. The control sleep was 8 hours in bed, and the restricted sleep was 5 hours, 4 hours in bed. Importantly, those where the restricted sleep was delayed, so they were up until late in the night. Restricted sleep subject gained 1 kilo over 5 days, while those who were in the control condition gained only 110 grams. African Americans gained more than Caucasians, and men gained more than women. Sleep-restricted subjects consumed 130% of their daily caloric intake, and mostly due to more meals. And meals I'm putting in quotes because really it is, some of these meals are really snacks, and that's, they're not main meals. Sleep-restricted subjects consumed more than 500 calories in the late evening and wee hours of the morning. And a number of these subjects participated in 2 studies, and their response, their metabolic response to sleep restriction was remarkably stable. So there are large and stable phenotypic differences in weight gain and evening eating in response to sleep restriction, particularly in males. So this is the large body of evidence from laboratory studies where we have looked quickly at the findings from 50 to 60 laboratory studies, has been complemented, of course, by a very large number of epidemiologic studies. This continues. Every epidemiologist who has access to a cohort where sleep was measured, usually by self-report, is looking now at metabolic measures, and the findings have been very consistent. There are more than 200 studies out there from around the world. But this meta-analysis is pulling the results of only prospective studies. So studies that examined short sleeper participants at baseline and then looked at incident obesity over the follow-up period. And the result of this statistical meta-analysis is that short sleepers have a 45% increased odds of developing obesity. Now, moving on to diabetes, for which obesity is the main risk factor, the same maps have been produced by the CDC and have that similarity again. And here, I feel that since it's probably the last time that I have the opportunity to give such a big lecture, I want to acknowledge our initial sleep debt study, which was published in 1999 in The Lancet, has an enormous number of citations, well over 1,000, and was led by Karin Spiegel and Rachel Leproux, who are wonderful colleagues and great friends. I want to reassure you that if you have a paper that is being rejected over and again, you should never take no for an answer and continue to try to publish it in the best possible journal, because this paper was rejected 4 times and is now one of the highest quoted paper in all of the field of medicine. So the sleep debt study was the first laboratory study of sleep restriction. It looked at 6 days of 4 hours per night versus 7 days of 12 hours per night. You may have heard the story, and the measure of glucose tolerance that we used is the intravenous glucose tolerance test, which was per IV GTT on this slide, which was performed after 5 nights of sleep restriction and 5 nights of sleep extension. And in this test, glucose, so there are a few baseline samples that are being taken. There are 2 IV lines, 1 for injections and 1 for sampling. At time 0, a bolus of glucose is injected. And at times 19, a bolus of insulin is injected. And you can see the decline of glucose levels is much slower when people were sleep deprived than when they were fully rested. And so that translates quantitatively into a decrease, a very significant decrease in glucose tolerance. At the same time, you can very well see that the initial response to insulin, the so-called acute insulin response to glucose, was decreased. And then what you cannot see are the 2 next parameters, because they are extracted from mathematical modeling of these insulin and glucose data. But they are showing that non-insulin dependent glucose utilization, which is mostly brain glucose utilization, is decreased. The tired brain is not using as much of its fuel. And lastly, that insulin sensitivity, which is really how much insulin is needed by peripheral tissue to metabolize the same amount of glucose, well, this insulin need this insulin sensitivity is also decreased. And taken together, these data indicated that after 1 week of 5 hours in bed, these young, healthy, fit men were in a pre-diabetic state. These findings have been replicated in a large number of studies. I'm only representing studies on this slide that are actually using the IVGTT and analyzing the data with the same mathematical model. And you can see that the results are very consistent in terms of a decrease of insulin sensitivity. The amplitude of the impact is between 20 and 35%. Studies that have only fragmented sleep, either eliminating slow wave sleep or fragmenting all sleep stages, have also, without changing sleep duration, have also seen a decrease in insulin sensitivity. So both sleep duration and sleep quality modulate insulin sensitivity in a very clinically significant way, because a 25% decrease of insulin sensitivity for a person of my body build would mean that I would put on 20 kilo. So, and that of course would be a disaster. We wanted to have, because at the time of when we did these studies, sleep, the motto in the sleep field was very much that sleep was for the brain, not for the rest of the body. And so we wanted to look at insulin sensitivity in a peripheral tissue that is relatively easily accessible in a human subject. And that tissue is is subcutaneous abdominal fat. And so Josie Broussard, who was in my lab at the time, proposed to collect fat biopsies in individuals who were participants in a randomized crossover design of short sleep, 4.5 hours, versus normal sleep, 8.5 hours. And then Josie isolated the adipocytes and put them in culture and exposed them to one of their major ligands, insulin, and examined as a response to exposure to insulin, an early step of the intracellular insulin cascade, which is the phosphorylation of AKT. So when the adipocytes were exposed to increasing insulin concentrations and the subject had normal sleep, you can see a clear increase of the phosphorylation of AKT as the dose of insulin is increased. When the subject were sleep restricted, at 0.10 of insulin there was no response, 0.25 there was no response, 0.50 we started to see some phosphorylation of AKT, and so that entire curve was lower and right shifted, meaning really that a state of cellular insulin sensitivity had developed in these adipocytes. So does fat need sleep? Yes. Fat needs sleep to function properly. And in fact, the effect size of sleep restriction is on cellular insulin sensitivity is very large. The final question regarding the impact of insufficient sleep is, of course, whether extending sleep in chronic short sleepers can have beneficial effects. And there are a number of studies that are ongoing and some are completed. But in this talk, I can only show published data. And I'm proud to say that it is Rachel Leproux, one of the co-authors of the sleep debt study, who published the first finding of a sleep extension study. And what she did is that she recruited healthy adults, but there was one inclusion criteria, is that the subject had, the potential participant had to wear a wrist activity monitor for 1 to 2 weeks, and they had to indeed being short sleepers, but extending sleep during the weekend to show that they were really sleep restricted by curtailing bedtimes rather than having some type of insomnia where they were unable to sleep. And here are the results. So you're looking at weekdays and weekends. She asked the subject to increase their time in bed by 1 hour, and they did that on average significantly during the 3 weeks of the intervention. During the weekend, they were already sleeping more, so they did not increase their sleep time during the weekend. And she examined fasting levels of insulin resistance and insulin sensitivity. And what the data are showing here is that there is a correlation between how much sleep extension was actually obtained, because everything was measured by wrist activity monitoring, and the decrease in insulin resistance, the top panel, and the increase in insulin sensitivity, the lower panel. So these data suggest that in a substantial fraction of chronic short sleepers, it is possible to extend sleep modestly and obtain derived beneficial effects on insulin sensitivity. These are best seen as preliminary data for a larger clinical trial, but they at least provide the base for further studies. So in front of this audience, I could not pass the opportunity to also discuss obstructive sleep apnea, sleep disturbances. Obesity and diabetes are interacting epidemics because obesity promotes obstructive sleep apnea. Obstructive sleep apnea promotes diabetes. Naresh Punjabi was an hour ago on the stage, and he really led extremely good work showing the impact of obstructive sleep apnea independently of body adiposity on insulin resistance. And I don't need to explain what obstructive sleep apnea is. I like to remind you that as a result of the epidemic of obesity, the prevalence is much higher, and it's much higher particularly in women, because while it was a rare disorder in women now, in women older than 50 years old who are obese, which represent a sizable portion of the population, the prevalence of OSA is 40%, according to this Wisconsin study. So when diabetes is already in place, then the prevalence of OSA becomes extraordinarily high, consistent with the adverse metabolic effects of sleep disturbances and also of hypoxia. And on average, from these 8 studies, the prevalence is 65%, which means that 2 out of 3 patients who come to see their GP or their endocrinologist for their diabetes, 2 out of 3 have OSA, and usually it's not diagnosed. OSA, the presence of OSA makes it more difficult to control glycemic levels with the dose-response relationship between the presence and severity of OSA and hemoglobin A1C, a marker of glucose control in diabetes. This data would suggest that CPAP would be a very useful anti-diabetic treatment in diabetic subjects with OSA. Unfortunately, several clinical trials have had disappointing results. They had one thing in common, is that compliance was very poor, with 3 to 5 hours for 4 to 5 days a week. I mean, I really wonder what drug would be tested for half the dosage, half of the time. But that's the way it is. But there have been some proof of concept studies in the laboratory that have had good results. And this is the study that was led at the University of Chicago by Babak Moklesi. And here we decided to have a proof of concept study. The subjects were going to sleep in the laboratory every night, and they would be outfitted with the best possible device. and every leak will be repaired, and every hand will be held, and the subject would therefore have 100% compliance throughout the night, all night. And so here's the 24-hour profile of glucose before and after CPAP, and you can see that the black curve, which is with CPAP, only 1 week, very remarkable drop of glucose, clinically significant. To achieve that kind of decrease, you would need one more anti-diabetic drug. And I was delighted to see that just recently the Hopkins group, led by Polotsky and John, used the model of CPAP withdrawal in a group of subjects that included diabetics, but the data are from the entire group. And you can see, if you focus on the glucose levels, that CPAP withdrawal resulted immediately in an increase of glucose levels, and it was mostly driven by the participants who were already diabetic. So this is, again, a totally controlled study, and what the field really now needs to do is to find, to tailor OSA treatment to the condition of the subject, but nonetheless really try to diminish the severity of this serious metabolic condition. So I've discussed the role of sleep disturbances for the risk of obesity and diabetes. What do we know about the role of the circadian system for metabolism? And I had mentioned already that the peripheral clocks can be affected by the timing of food intake. And the first data that supported a strong relationship between the circadian system and metabolism were actually obtained in the clock mouse, the clock mutant, and published in 2005 by Frederick and Joe Bass. And Joe will be presenting tomorrow, and you will have a lot more details about this. But the clock mouse mutant, while it's born normal weight, as it ages, it puts on a lot more weight than the wild type, particularly if it's fed a high-fat diet. So it is a metabolic phenotype that is also associated with the development of type 2 diabetes at an earlier age, and with a fatty liver. Bass loves to show disgusting photos of the liver of the clock mutant. I'm sure you can see it tomorrow. And his subsequent work, though, is very elegant and demonstrated a pivotal role of the circadian clock for metabolism. And here I am going to describe this very cautiously because, as I mentioned, my biochemistry and molecular biology background is minimal. But the BMAL clock complex drives not only the expression of purr and cry, and also nuclear receptors, ROR and REV, but also drives the expression of NAMPT. And NAMPT is a rate-limiting enzyme for the production of NAD+. And NAD+ is a cofactor that is necessary for two important things. One, it is necessary for absorption of nutrients and transformation of nutrients into cellular energy. And two, NAD+ is a must for the sirtuin family of genes to be active. The sirtuins are described as the genes of the fountain of youth. These are genes that are activated by caloric restriction, which extends longevity, as you know. But also the sirtuins have the capacity of repair DNA. And so the sirtuins link the circadian clock to aging, and by NAD+, and NAD+ also has the ability to promote the absorption of nutrients and transform them in cellular energy. So this molecular model, which Joe and his collaborators studied mostly in the liver, is now well documented. And NAD+, because it both gives more cellular energy and is anti-aging, is becoming one of the most, the best sellers of nutritional supplements. There is a precursor of NAD+ that is orally available and sells like hotcakes. I have bought one myself, bottle myself, but I have to still really carefully think about when to take it, because of course, since the whole system is oscillatory, you don't want to take it when it's going to counteract the normal circadian variation. So besides these, very strong and remarkable molecular links, in terms of human data, there is not that much. Of course, there is the epidemiology of shift work. And here, this is a slide from the Nurses' Health Study showing that there is a graded relationship between the years of shift work and weight gain. This stops for nurses that are more than 20 years in shift work, but as my colleague Kristen Knudsen has explained to me, this is a survivor effect, that only people who remain in shift work for more than 20 years are kind of tolerant to its adverse effects. There's also a lot of data, epidemiologic data, linking shift work to the risk of incident diabetes, so you're more likely to develop diabetes if you are a shift worker. And this is a meta-analysis where shift work was shown to increase the risk of developing diabetes by 40%. Laboratory studies are few, and it's important to recognize that it's not a simple project, because in shift workers, the sleep is always diminished as well. So all of this epidemiologic evidence linking shift work to the risk of diabetes and obesity is partly contaminated by the fact that shift workers always sleep less, and so there is also a sleep debt. And in laboratory studies, there's been an effort to control for that. The first study that did control for that statistically was led by Frank Scher at Harvard Medical School and used the 28-hour forced desynchrony protocol, and examined glucose and insulin when the subject were 12 hours out of phase with the normal circadian time. And you can see that there was a marked increase in both glucose, and this was clinically significant. We conducted a study where we controlled experimentally for sleep duration and observed that the decrease of insulin sensitivity is essentially doubled by circadian misalignment despite identical durations of sleep. So this is my final slide, so we're almost done. We did a study comparing the risk of diabetes, of developing diabetes, as compared to the traditional risk factor that each and every statement of the American Diabetes Association lists in the recommendations to practitioners. And these are being overweight, which increases your risk 3 times, family history of diabetes increases your risk 2.4 times, and physical activity, which has a modest increase in risk at 1.15. And if you look at the sleep and circadian disturbances, including OSA, which overlaps completely having a family history of diabetes, yet the majority of patients are not even diagnosed, difficulty maintaining sleep, difficulty rotating shift work, difficulty initiating sleep. All of these error bars overlap the risk associated with the genetic background of the subject. So why is it that these risk factors are never mentioned, and that our physicians are not even asking their patients, are you a shift worker? So this is where precision medicine, based on taking into account the genetic background of the individual, if it doesn't take into account the circadian history and the sleep history, it really is not called precision, in my view. So I want to conclude by saying that an enormous amount of data from epidemiologic study, animal models, clinical research has shown unequivocally that sleep disturbances and circadian disturbances are risk factors for these major diseases. The Nobel Prize gave a stamp of approval and validity to the study of sleep and circadian rhythms. We must take on the challenge in a way and the momentum that this award has given to our field. My last slide shows the collaborators that are still tolerating me at the time. And I was thinking of putting a slide together with all the people that I have quoted in this talk, but it was just overwhelming. So I'm thanking them for continuing this work, and thanking you very much for your attention. Merci beaucoup.